Hi there, my name is Ryan Dickerson. I'm a medical student in Brody School of Medicine class of 2025, and this is our video on fertilization and implantation. This video is brought to you by the Scrubs team, and Scrubs is a student-driven initiative that aims to develop supplemental resources for current and future cohorts that pass through Brody. Here's our mission statement that you are welcome to view. Quick disclaimer for all of our resources. All of our resources are made as supplemental resources and in no way take the replacement of Brody faculty. We encourage you to use these resources with this in mind. With that, let's go ahead and get to the content. Before we get too far into fertilization and implantation, there are some terms that I'd like to identify just to make sure that we're all on the same page moving forward. Firstly, homologous chromosomes. Homologous chromosomes refer specifically to chromosomes that are in pairs in a diploid organism. So this means that one chromosome is inherited from each parent. So you can see here we have homologous chromosomes with one chromosome inherited from the male, the other chromosome inherited from the female. Even after replication, these chromosomes are still homologous pairs. That differs from a sister chromatid. A sister chromatid refers to a single chromosome that has been replicated, and each one of those components is known as a chromatid. Together, that paired version is known as a sister chromatid. So this is due to each duplicated chromosome consisting of two separate chromatids that are joined at the centromere. Another term that's going to be helpful to identify is diploid versus haploid organisms. So a diploid cell contains two complete sets of chromosomes. This means that it's containing one from each parent. So in this case, this cell would be a diploid cell. A haploid cell, however, contains only one set of chromosomes, which we'll see happens after meiosis 1. The key difference in terms, because we're going to be using these throughout this video, is that a chromosome versus chromatid, the chromosome is a single structure, while the chromatid could be one of the two halves of a replicated chromosome. So you can see that here, these are sister chromatids, whereas each one of these is a chromosome as a whole. Next thing is when you refer to a chromosome versus homologous chromosomes, the chromosome is a single structure, whereas the homologous chromosomes refer to a pair. And so these would be homologous chromosomes, meaning that there's one from each parent. Because if we just say chromosome, that's only referring to one of these structures, not them as a pair. So be careful to make sure that you are listening for if we're using the term pair, homologous, chromosome, chromatid. All right, with that, we're going to keep on moving forward. So in order to understand embryology, we first have to understand a little bit about meiosis. And this is just going to be a quick, brief overview. The big thing about meiosis is broken down into two stages. We have meiosis 1 and we have meiosis 2. The goal for meiosis 1 is really to segregate our chromosomes. So the goal here is to take the homologous pairs and separate those. So we're going from a diploid organism to a haploid organism. Okay, so just meiosis 1, meiosis 2 are very similar. We're going to have interphase, prophase, metaphase, anaphase, and telophase. Telophase being the eventual separation of the cells. However, meiosis 1, this is the component right here in telophase that we separate from the diploid organism to the haploid organism. So that's really important that you know after meiosis 1, you are now at the haploid stage. During meiosis 2, now we're going to go ahead and separate the sister chromatids from one another. So the sister chromatids are going to line up in metaphase. Then we're going to separate those in anaphase, and then in telophase, divide those cells. So now we end up with 4 different gametes. And the gametes are essentially going to be the building blocks of the cells that we'll have for replication and for embryogenesis. So in understanding how these cells are going to form, we initially have to think about spermatogenesis, and this is the formation of the gametes in the male. So spermatogenesis occurs entirely within the testicle in males, and these sperm, once they are formed, are stored in the epididymis. So after spermatogenesis, you have 4 sperm that arise from a single cell, and each one of these has 23 single chromatid chromosomes, and they're stored in the epididymis. During ejaculation, the stored sperm that are in the epididymis are going to pass through the vas deferens. They're going to come up through and they're going to have contributions from the secondary sex glands. So this will be your seminal vesicle as well as your prostate. They're going to add secretions to form semen, and the semen is going to pass through the urethra and then out of the bulb of the penis, the glands of the penis, into the female reproductive tract for fertilization. So the big point here is that Spermatogenesis occurs entirely within the testicle, and then it's stored in the epididymis. Eventually, sperm is going to progress out of the male reproductive system through the gland's penis into the female reproductive tract to allow for fertilization. Now, while sperm may pass out of the male reproductive tract during fertilization, this does not mean that those cells are ready to fertilize an egg. In order for that, there has to be a maturation stage, and we call this capacitation. So this requires approximately 7 hours of being present within the female genital tract, and it's because the female genital tract releases different enzymes that are going to help activate the sperm. So 3 things happen during this capacitation phase. 1, you remove the glycoprotein coat and seminal proteins that are on the acrosome of the sperm. And the acrosome is really the head of the sperm here. There's going to be different enzymes that are going to be contained within that, which we'll talk about later, but these are really proteolytic enzymes that help break down proteins that are surrounding the female gamete to allow for fertilization. So step 1 is removing the glycoprotein coat, which you can see here. The second is there's going to be a decreased membrane cholesterol and phospholipid ratio. And lastly, hyperpolarization of this acromial membrane, and that is just to prevent that early release for all of those proteins to make sure they're not released prematurely. So again, this is the capacitation stage of sperm, allowing them to be motily active in order to fertilize the egg. Now, while we talked about meiosis 1 and meiosis 2 occurring naturally in males, just as you've learned previously, the production of gametes in females, known as oogenesis, is slightly different. The big thing here is that in development of your gametes, all of the eggs for a female are going to be arrested as primary oocytes in prophase 1 sometime before the start of menses. Okay, so all of those primary oocytes are arrested in prophase 1, so right at the beginning of meiosis. And at this point, they contain those 46 double chromatid chromosomes, because they haven't gone through meiosis 1 yet, so that means that they haven't gone from haploid to the diploid stage. Now, under the guidance of female sex hormones, primarily progesterone, estrogen, and specifically the LH surge, you're going to have that LH surge trigger ovulation, which is going to cause a primary oocyte to be released from the ovary, which is the female gonad, and then that's going to progress into the secondary oocyte. So the primary oocyte gets released. That causes the progression of meiosis, which will eventually pause once again in metaphase 2. So the progression goes from prophase 1 all the way to metaphase 2. So at this point, you've now transitioned to 23 double chromatid chromosomes. So you're at the diploid stage. Now what you'll notice here is that you see that you form a secondary oocyte, but then you're also giving off a polar body. What this is basically saying is that cell divisions within oogenesis are unequal. So you're going to have one larger cell that's going to be released containing most of the organelles like mitochondria and other things that help the cell function, whereas the other division is going to be smaller and it's not going to be helpful for development of an embryo. And this is really to make sure that you're having the biggest end egg possible, so that way you have the nutrition available for initial stages of development. So now these secondary oocytes, they've been released and they're now going through the fallopian tubes to meet the sperm. They're again arrested in metaphase 2 until that point. Once sperm enters the cell, they will complete meiosis 2, and then they are going to be known as the ovum. So this is the production of gametes for females, which is also known as oogenesis. Now I do want to point out, as we're talking through this, it might be helpful to review a little bit about the female reproductive system. Inferiorly, we have the vaginal canal, which is going to rise superiorly to the cervix. Then we have the body of the uterus. The important thing to note is that the inside of the uterus is going to be known as the endometrium. So that term is going to be coming up later. The endometrium is connected to the fallopian tubes. The fallopian tubes are then going to go distally, and you're going to have the infundibulum, which is this opening, the fimbriae, which are little finger-like projections of the fallopian tube, and then the ovary. So remember that the female gametes, the primary oocytes, are found within the ovary. During ovulation, which is caused by the surge in LH, is going to be the primary oocyte is going to be released. It is going to progress into metaphase 2 as a secondary oocyte, and it's going to be floating along, hopefully caught up by the fimbriae into the fallopian tubes. And then the site where it is most often fertilized by the sperm is going to be the ampulla. So the ampulla of the fallopian tube is a site where you're most often going to have the secondary oocyte fertilized by the sperm. Now the other thing here, you see this number 2, this is indicating that the most common location and the expected location for implantation is going to be within the endometrium of the uterus. Anywhere outside of that is going to be known as an ectopic pregnancy, which we'll talk about at the end of this video. Okay, so now we've got our gametes, but we need to fertilize in order to form our eventual embryo. So this means that we need the combination of both male and female components. So the process for this goes through a couple of different stages. Initially, we're going to have the sperm and secondary oocyte meet the ampulla, which we talked about is the most common spot for fertilization to occur. Once this happens, the sperm have already gone capacitation, so they're ready to fertilize the egg. Now, in order to do that, the egg is surrounded by 2 different layers. One external layer is called the corona radiata, and then more proximally we have the zona pellucida. So those are the 2 layers that surround the egg, and what this is doing is preventing sperm from actually entering to fertilize, meaning that because there's millions of sperm, only one is going to be able to end up doing this. So in order to make it through these different layers, the sperm actually undergo the acrosome reaction, and this is going to be all of those enzymes that are present within the acrosome of the sperm. They are going to release different enzymes such as hyaluronidase and acrosin. These are just different enzymes to break down proteins that are found within the corona radiata. So once the sperm are able to penetrate the corona radiata, this is going to then, the sperm are then going to bind to the zona pellucida, and this is going to cause something known as the zona reaction. So this zona reaction is mediated by a bunch of different chemicals, which we're not going to go into. But the goal here is that the zona reaction pretty much forms a wall that no other sperm can cross. So as soon as the very first sperm that hits the zona pellucida is bound, the zona reaction occurs, meaning that no other sperm will be able to penetrate the egg. And this ensures that there's only one sperm fertilizing the egg as opposed to multiple sperm. Now, once this happens, the sperm is actually going to enter the secondary oocyte, and it's going to fuse. So the cell membranes are going to fuse together. And this is going to trigger meiosis to complete within the secondary oocyte. Now, the nuclear membrane will break down, and when this happens within the secondary oocyte, it's now going to be known as the female pronucleus. The male nucleus is going to do the same. It's going to enlarge. It's going to break down its nuclear membrane, and that's going to be known as the male pronucleus. When both of these pronuclei are present within this cell, the cell is now known as an otid. Okay. The combination of these pronuclei, when they come together and they form a new nuclear membrane, which now has all 46 chromosomes, 23 from mom, 23 from dad. This is now a diploid cell, and it's going to be known as a zygote. All right, so the overview of fertilization, the results that you now have is you have the zygote formation, which is a diploid cell, has 46 chromosomes. If the sperm that enters the egg contains X chromosome, this is going to derive a female embryo. If it is a Y chromosome, it is going to derive a male embryo. Now, as soon as fertilization occurs, cleavage is going to be initiated, which is what we're going to talk about in this next step. So for cleavage and compaction of the embryo, this happens after fertilization. It takes about a day after, and then you're going to start to have cell divisions. So these cell divisions are going to happen rapidly. And this is going to go from the zygote phase. We're going to have multiple cell divisions, and once we hit the 32 cell stage, we start to call it a morula because it resembles the size of a blueberry. Now, the thing here is these divisions occur asynchronously, meaning that we're not going to have synchronous divisions. We're not going to have a cell that looks exactly symmetric. So you're going to have an asymmetric embryo that's going to be forming. Now, these cells, once they start dividing, instead of being called a zygote, they're going to be called blastomeres. So these individual cells. So every single one of these cells you see is a blastomere. Now, eventually, around day 3, when we're around the morula stage, or just before, we're going to have something called compaction. And this is when the blastomeres are going to adhere tightly to one another. And when this happens, you're going to form an inner layer and an outer layer. This is going to be important as you'll see in just a moment. So again, this happens around day 3. We have the morula phase. We've now gone through the fallopian tube. We're now getting ready to go into the uterus. Once we enter the uterus, there's a lot of uterine fluid that will be present. That uterine fluid is going to force open that morula, and it's going to form a space inside. That is known as the blastocystic cavity. Okay, the space here. It's also going to form different cell layers. We're going to have an inner cell layer, which we can see here, and we're going to have an outer cell layer, which is surrounding everything else. The inner cell layer is known as the embryoblast. Okay, inner cell layer known as the embryoblast. This outer cell layer, which is surrounding the inner cell layer and also contains the blastocystic cavity, this is going to be known as the trophoblast. Right? This structure, when it forms as a whole, is known as the blastocyst. So once more, the blastocyst is going to have an external layer known as the trophoblast and an internal layer known as the embryoblast. Within the trophoblast, you're also going to have the blastocystic cavity, which is full of fluid. All right. Now, during this process, you see that there's a sheath that surrounds the morula at this stage, and it is eventually gone as we have the blastocyst stage. What's happening is we still have the zona pellucida that's surrounding the developing blastocyst, and it will eventually fall off about 2 days after you go from morula to blastocyst stage. So about 2 days later, you're going to have the zona pellucida fall off. The next step in this process, now that you've gone from the blastocyst formation stage, you're now in the uterus, is going to be eventual implantation of the blastocyst into the uterine wall. So this usually occurs around a week after fertilization. Remember, fertilization is most commonly happening at the region of the ampulla of the fallopian tube. It will take about a week for implantation to fully occur, so that way you have good adherence to the developing embryo and the uterine wall. The specific name of the uterine wall structure that we're implanting into is going to be the endometrium. The endometrium is that single layer lining that is on the internal surface of the uterus. So that's going to be where we're going to be implanting. So in order for the implantation process to occur, there's a couple of different steps. First thing that's going to happen is going to be invasion and differentiation of the trophoblast layer. Remember, for our blastocyst, the trophoblast was that external capsule, whereas we had the internal embryoblast along with the blastocystic cavity. That trophoblast is going to then differentiate into 2 layers. One is going to be the cytotrophoblast, which is going to be the internal layer, and then we're going to have the syncytiotrophoblast, which is going to be this external layer. So the cytotrophoblast is the inner layer of mononucleated cells, meaning that they only have one nucleus per cell. And these cells are mitotically active, and their major goal is to provide new cells to contribute to the outer layer, which is the syncytiotrophoblast. So the syncytiotrophoblast, on the other hand, is a layer of multinucleated cells, meaning that there's multiple nuclei per cell. And this is because they arise from the fusion of these cytotrophoblast, and they lack distinct cell boundaries. So you can actually see in this image here there are multiple nuclei in this one cell. So I'm going through the different functions of the cytotrophoblast, which remember is that internal layer of the trophoblast, and then the syncytiotrophoblast. The cytotrophoblast is primarily the source of cells for the expanding syncytiotrophoblast, and it also is going to help maintain structural stability for the developing placental tissues, because remember the trophoblast's primary goal is going to be to develop into the future placenta, which is the interface between maternal blood supply and fetal blood supply. The syncytiotrophoblast is what's actually invading into the uterine wall. So the invasion is into the endometrial tissues, and this allows the blastocyst to embed itself deeply into the uterine wall, and it does this by releasing enzymes to aid in the process. Okay, but the other thing that we have for the syncytiotrophoblast is also going to be producing hormones, and the most important hormone for us to know is human chorionic gonadotropin, or also known as beta-HCG. And this is crucial for maintaining the corpus luteum, which is secreting progesterone, and this is enabling the uterine wall to be supported in pregnancy. And this is actually the hormone that is tested in the home pregnancy test. The last thing that's important to know about the syncytial trophoblast in terms of function is that it's also necessary to facilitate nutrient and gas exchange between maternal and embryonic blood supplies. Because remember, the end goal is to develop into the placenta, which is going to allow for that interface to occur. Now, as the syncytial trophoblast continues to invade, eventually you're going to have spaces that start to form. These spaces are called lacunae. And within these spaces, they're going to fill with maternal blood. And as the syncytial trophoblast continues to erode into maternal capillaries, the maternal blood flow is going to fill the lacunae, and it's going to establish that primitive uteroplacental circulation, which is going to really be the interface between fetal blood supply and maternal blood supply. So again, the end goal here is to develop into a placenta and to develop that interface between the two blood supplies. And we do that because the trophoblast is again dividing during the implantation phase into the cytotrophoblast, which is the internal mononuclear cell layer, and the syncytial trophoblast, which is the more invasive multinuclear cell layer. Okay, I know that was a rapid overview, but now we're going to go through a couple of clinical anatomy points, and then we'll wrap up this video. So, the first point I want to make is aneuploidy versus polyploidy. So aneuploidy refers to a chromosomal abnormality where there is one extra or one missing chromosome. So this is one chromosome specifically. And the most often cause of this is going to be non-disjunction. It's really important that we know that the most common cause of aneuploidy is going to be non-disjunction of either meiosis 1 or meiosis 2. Now if you have only one copy of a chromosome, whereas while you're supposed to have two, if you have one copy, it's called a monosomy. If you have 3 copies of the same chromosome, that's going to be known as a trisomy. Now, some of the most common trisomies that you're going to need to know are going to be trisomy 21, trisomy 18, and trisomy 13. These are known respectively as Down syndrome, Eber syndrome, and Patau. You'll learn about these later in your studies, but for now I want to point out trisomy 21's definitive features involve the Simeon-Palmer crease, a flattened nasal bridge, a smooth philtrum, which is the area right above the upper lip, mental delays, and cardiac defects. Trisomy 18 and 13 both have features that are important to recognize on test. However, for this video, we're just going to say that often these cases are fatal within their first year of life, and most other trisomies are not going to support life. Now, those are aneuploidies, meaning that there's one chromosome abnormality. That is different from a polyploidy. A polyploidy refers to an entire set of chromosomes that are missing or added in addition. So this is the whole set of 23 chromosomes. An example for this, when we have an extra set of 23 chromosomes, is known as triploidy. And the most common cause of this is fertilization by multiple sperm. And remember that the way that the body protects against this is because we have the zonal reaction, which causes the zona pellucida to harden, almost like concrete, to keep other sperm from invading the secondary oocyte, causing fertilization by multiple sperm. Right. And here's an example down here. If you just need to think back to what it means for there to be non-disjunction, the big point here is that non-disjunction can happen in meiosis 1 or meiosis 2 that can eventually lead to one of these aneuploidies. Now, aneuploidies of the sex chromosomes are a little bit specific because they're going to present differently than trisomy 21, 13, or 18. So a monosomy for a sex chromosome that leaves you with just one X chromosome is known as Turner syndrome. So this is 45X or Turner syndrome, and it has a couple of different features. Because you have the X chromosome, you are going to be phenotypically female. There's no Y chromosome present, but you often are going to present with short stature, a webbed neck, and what is often described as a broad shield-like chest. He has a very broad chest, and often no secondary sex characteristics will develop, meaning that you may not have the breast enlargement that you would normally see with puberty. 47XXY, so this is a trisomy. You have an extra set of the sex chromosome, specifically have an extra X. This is going to result in Klinefelter syndrome. These individuals, because they do have the Y chromosome, are going to be phenotypically males. They often are going to present with hypogonadism, or small testes in this case, and they often may present with gynecomastia due to extra estrogen that's present in their system. They often have long legs, so long legs with tall stature. That is Klinefelter syndrome. The last one is double Y males, so now you have a aneuploidy or a trisomy of an extra Y chromosome. These are going to, because the Y chromosome is present, phenotypical males. They are tall in stature, and they are aggressive in behavior most commonly. And for the last clinical correlation that I want to talk about, second to last, we're going to talk about ectopic pregnancies. An ectopic pregnancy refers to any time that there is implantation, so not fertilization, but implantation of a fertilized egg somewhere outside of the uterine cavity. Again, this is supposed to be the correct spot of implantation, but if it happens within the fallopian tube, if it happens within the ovary, if it happens outside of the ovary fallopian tube or uterus and is in the abdominal cavity, this is also known as an ectopic pregnancy. Rarely, you can also have an ectopic pregnancy that is implanted too deep within the uterine wall, not within the endometrium, but within the myometrium or the musculature layer. So again, ectopic pregnancy refers to any pregnancy where implantation occurs outside of its normal variation within the uterus. And for our last clinical correlation, really this is just defining the difference between identical twins and fraternal twins based on how cell division occurs. For identical twins, this is going to be due to a single egg and a single sperm that are then going to split in two after fertilization has occurred. Okay, so this means that they are going to be identical in sex, they're going to be identical in features, they're going to have the same genetic code. Fraternal twins, however, most often occurs due to this, two different eggs being fertilized by two separate sperms. Okay, or two different sperm. And this is going to mean that you share only half your genetic code, or basically you are going to be siblings, and just like any other siblings that are spaced by years, because there are two separate eggs here that are going to be fertilized by two separate sperm. All right. So really recognizing that identical twins are going to be caused by fertilization and then separation, as opposed to fraternal twins, there being separation and then fertilization secondarily to that. And with that, that'll bring an end to this video.